Hello, everybody. This is the Chapter 17 lecture on osmoregulation. To begin, we'll look at the structure and function of the kidneys. The kidneys play a key role in homeostasis, specifically in regulating the extracellular fluid in the environment, specifically the volume of blood, which can have impact on blood pressure, waste, electrolytes, which can also affect blood pressure, and pH. So urine is made in the kidney and it drains into the renal pelvis, then down the ureter to the urinary bladder. Then it passes from the bladder through the urethra to exit the body, and it is transported using peristalsis. And of course, we know that is a process involving the contraction of smooth muscles. So here is a diagram showing some of the organs of the urinary system. The key organs include the kidneys, which we have two, and the bladder. So if we take a closer look at the structure of the kidney, we can see the kidney has two key regions. One is the renal cortex, which I'm pointing to with the pointer, and then deeper in the kidney, we have the renal medulla. And the renal medulla is made of renal pyramids, which you can see these triangular structures, and also renal columns that are in between the renal pyramids. And each renal pyramid drains into a minor calyx, and then that goes into a major calyx, and then into the renal pelvis. So here we see one renal medulla, and this can drain into the minor calyx, and then drain into the major calyx after that. And you can see the major calyx over here. The minor one is at the top. So the nephron, which is what we see down here, this is the functional unit of the kidney. And each kidney has more than 1 million nephrons. The nephrons consist of small tubules and associated blood vessels. And the blood is filtered first, then it enters the tubules, becomes modified, and then leaves the tubules and is collected as urine. So I'd like to point out some of the key regions of the nephron. So first, we have the glomerulus, which is over here. Another name for this is called Bowman's capsule, or the glomerulocapsule. And this plays a key role in filtration. And what I want to show you what we have interacting with this is an afferent arteriole, and this is carrying oxygenated blood, and it basically will be put into this little capsule, and then the pressure from the blood will help to, along with some other factors, will help it to be the fluid to be pushed through the filtration apparatus of the glomerular capsule. And this plays a key role in filtration. The next stage for the filtrate is to go through the proximal convoluted tubule, which I'm showing you right now. The job of the proximal convoluted tubule is to play a key role in reabsorption of water. Then we have the loop of Henle, a descending loop, an ascending loop, and the descending loop of Henle, which is the first portion, the descending loop of Henle, its job is concentration. And the ascending loop of Henle also plays a role in concentration as well. So both the descending and ascending loop of Henle play a role in concentration. And then we have the distal convoluted tubule, and its main function is secretion. And then the filtrate can drain into the minor cowlicks after this. So I just want to reiterate the four regions of the nephron and what they do. So the Bowman's capsule, its main job is filtration. The proximal tubule, its main job is reabsorption. The main job of the loop of Henle is concentration. And the main job of the distal convoluted tubule is secretion. And we'll be going through these in detail in this lecture. So there's an osmotic gradient that occurs in the nephron, and specifically the most interesting part of the osmotic gradient is occurring in the loop of Henle. So I want to show you that in the beginning, so another thing to mention is that the beginning of the loop of Henle has an osmolarity of about 450, as you can see over here. And as you go up, I want to point out that the loop of Henle is completely located in the medulla. Yes, loop of Henle is completely located in the medulla. The proximal tubule, convoluted tubule, and the distal convoluted tubule transverse both the cortex and the medulla. So I just wanted to highlight that to you. And the osmolarity in the cortex is the normal 300 millismol, which you find throughout the body. But as you go deeper in the medulla, the osmotic gradient increases all the way up to about 1400 milliosmoles at the bottom of the loop of Henle. And in the loop of Henle, as we're going to talk about this more, this osmotic gradient will play a key role in water leaving, because the water is going to be following the solutes that are in the medulla, the renal medulla, and then in the thick. So, and by the way, I should mention the descending loop of Henle is permeable to water, but it is not permeable to potassium, sodium, and chloride transport. And then, in contrast, the ascending loop of Henle is impermeable to water, but it has sodium, potassium, and chloride transporters, and it's actually going to be pumping them out into the renal medulla. And we'll be taking looking at this in more detail when we get to the loop of Henle, because we're going to look at each region of the nephron in greater detail. And then critically, if you look at the kidney, we see there are many arteries and veins that are throughout the entire kidney, as we expect for any tissue in the body. But of course here, they're playing a key role where the arteries are interacting with the Bowman's capsules to initiate the filtration process. And I just want to have this slide to remind those of you who like seeing pictures and learn visually that when we have the Bowman's capsule or the glomerule capsule, we have water leaving for the filtration, and also small molecules can also be filtered out of the blood vessels, including urea, glucose, amino acids, uric acid, salts. Proteins usually do not pass into the filtrate. And I just want to show that the movement of molecules in the proximal convoluted tubule, the loop of Henle, and the distal convoluted tubule are vast, and there's, so these are all small molecules, or the proximal just convoluted tubule. We have urea, uric acid, creatine, some drugs, glucose, amino acids, this huge list. You don't have to memorize any of this list. I'm just showing you to have you appreciate the diversity of solutes that we can find in the nephron, in the filtrate of the nephron. And we'll be talking about some of these in greater detail as we go through each one of the regions.